Hi, everyone. I just wanted to say welcome, and I wanted to thank Dr. Akhtar and Dr. Kadir for being willing to be here to talk with us. This is something that stumbling across Dr. Akhtar's TED Talk was very helpful to me when I was in residency and fellowship, and as an attending, to answer the question that I always get from people when I'm asked why I object to animal testing. So it's been very nice to know Aisha and to have her as a resource. So I'm super excited about what we'll.
Thank you so much, Priya, for having us, and Swati and Pratik.
Thank you, Dr. Priya. Thank you, Dr. Aisha. Dr. Kadir, over to you. Please share your thoughts, and thank you so much for being here.
Yes, thank you so much for inviting me on this call. I'm very excited to be here. I'm coming on as the Director of Science Innovation for Center for Contemporary Sciences, work directly under the leadership of Dr. Aisha Akhtar, so very proud and humble for me to be a part of the organization. Definitely would love to share with everyone a little bit about just animal use in the medical scientific space, and I'm sure we'll get into more discussions into that later on, but happy to just put in my input as a scientist and someone that's been in this biomedical space for a very long time. So thank you so much for having me.
Thank you, Dr. Kadir. Dr. Aisha, over to you. What do you?
No, I'm ready for the questions. I know Pratik and has some questions for us, so I say let's forge ahead.
Thank you so much, Pratik. Yeah.
Yeah, before we start, I would actually like to thank the entire team of Planet Earthlings, as well as the Global Jain Network, and a lot of people belonging to these teams are in the audience. So, like this wouldn't have been possible without the support of these wonderful people that we have over here, in the speaker panel, as well as in the listening lounge. Before Yeah, so Swati, I also know that you also have like a list of questions. So, but before that, maybe we can just take like a few opening comments, brief comments from Deep and Watch, and then we can just start with the questions. So Deep, if you want to go ahead.
Thank you for having me, Pratik. As a layman and a vegan and somebody who cares for animals, this is a really interesting subject, important subject. There are so many products that we consume, but we are unaware that these have at first been, tested on animals. And I'm just eager to learn if there are other testing methods possible and what we as consumers should do so that products that are tested on animals, do not have a demand in the market, and so that their supply decreases. So I'm eager to learn more on the subject. Thank you for having me.
Thank you, Deep, for that. Let's quickly hear from Vaj. Vaj, please.
Thank you so much. Thanks to Dr. Priya for bringing me in. Hey, Dr. Kadir. Hey, Dr. Aisha. Really great to see you. I'm really interested in the work with Center for Contemporary Sciences is doing, also following the lung on a chip model, which is based in line on the organ on a chip model, and also the interaction between the various chip models and the overall replication of the body as a whole. I'm not sure if there is any effort going on to coordinate these interactions between individual testing on organ on a chip model and doing an integration testing basically to simulate the whole function of the body. Also want to get more insight on the new vaccines which are going to be done for not only the Omicron but the HPAI or highly pathogenic avian influenza, if they can be simulated on an organ on a chip model. So really looking forward to this conversation. Back.
Thank you, Vaj. Really appreciated. Pratik, do you?
Yeah, thanks. And I've also pinned the link for the for Contemporary Sciences. So if you all just want to take a look at the work, the partners, the sponsors, it's all there on the website, and it's a great website. So to start with, I have like a few questions, but the first question is why? Like, why are animals being used for testing, or what's the purpose of this testing? So maybe Dr. Kadir, if you want to start with answering that question. So why are animals being used, and what?
Sure. So animals have been used notoriously in the biological sciences, biomedical science space, for a very long time, since inception of research entities being formed across the country and really on a global scale. We use them because, at least I can speak in part of what goes on here in the U.S. A lot of the thought process has been around whenever there is specific drugs that we would like to discover. So when we talk about drug discovery and development, disease modeling, or even as of recent personalized medicine, we have this thought that we need to investigate not only the disease of interest or drugs of interest um, in the lab space when it comes down to using cell culture tissues and stuff like that, cells and tissues, but also we need to make sure that we model it into a living species. And Dr. Aisha Akhtar will actually talk about this, I'm sure, during our conversation today, but in the Fda Modernization Act of 1938, it actually stated that in order for drugs to be approved, scientists, in fact, needed to use two different species of two different sizes, a small and a large animal. And we've been sort of following that practice, a very antiquated practice now for some time, for almost 100 years. And so the thought in the culture of research organizations and research institutions has been that in order for us to get drug approval, and then to push those drugs to the patient bedside, we in fact have to investigate it into animal models. What I know, and specifically what I know as a scientist who's trained in this space for decades, is that a lot of times animal models, the experimentations that we do are not reproducible. We don't get consistent data. We're sort of connecting outliers. In the Center for Contemporary Science LinkedIn Live interview that we had earlier this week on Tuesday with Dr. Akhtar, she actually framed it the best way, which, I could definitely back her statement, which was, we're trying to sort of simulate this disease model into an animal. So we're creating this disease into an animal, or we think we are, but what we're really doing is we're creating the symptoms of the disease, and then the scientists turn right back around and try to find ways to, deplete those symptoms in the animal. So there's just a lot of disconnect that exists in the research space, and we have been following, like I said, at least here in the United States, some antiquated practices within the Fda Modernization Act of 1938, saying that we in fact have to do that in order to get drugs approved. Unfortunately, a lot of the private industries and federal agencies obviously follow suit to what the FDA says, and the culture of those organizations are very difficult to shift their mindset when it comes down to using, something like human relevant testing methods and other alternatives that would actually, in fact, give us better data, more reproducible results, definitely more correlated to direct patient outcomes. And at the same time, we would not be using animals. We could replace animal use and create a more compassionate scientific environment. So that's really my answer. And I don't know if Dr. Akhtar wants to add anything else to what I just mentioned.
Hey, yeah, thanks, Dr. Kadir. So, Dr. Akhtar, do you also want to add? And before that, like, did Dr. Kadir say small and large animals? And does that mean for every particular test you're using like at least one species of smaller animals and one species of larger animals. Is that right? Or
Well, let me go ahead and just kind of break it down. So there are different categories for which animals are used. One is education. A lot of animals are used in high school classes and college classes, even elementary classes, right? For either dissection or experimentation, or they're used for training in some way in upper levels. Then there's a category called safety and toxicity testing. And this is where animals are used to test the safety of a chemical compound or a household a product like cosmetics or shampoos or industrial chemicals, agricultural chemicals. And as you can imagine, they're pumping these chemicals, these ingredients into animals, into their eyes, onto their skin, down their throat. I mean, you can imagine it's incredibly very painful. This category is very painful for the animals involved. Then, so that's probably about maybe 20% of the overall number of animals used worldwide. Now, what most people think about when they think about animal testing, besides like cosmetics and household products, is then drug development. Drug development is another category for which animals are used, and in that area, that's really the Food and Drug Administration in the US has oversight here. There's a regulatory agency that either allows for a drug to move from the testing stage to be tried in human clinical trials, and then allowing it to move further and to be approved for the market. Now, the Food and Drug Administration, with rare exception, will require every new drug and vaccine to be tested on at least 2 different species before going into human clinical trials. So there's usually one species is a smaller species like a mouse or rat. The other species will be a larger species like a non-human primate, a dog, or a pig. Don't ask me why they have that kind of format. It doesn't make sense, but that's nevertheless the way the FDA has functioned for decades, as Carmen has mentioned. Now, for drug development, animals are probably, well, in this category, we're talking probably about 25% of the overall use of animals in biomedical research. Now, what most people don't realize is that most animals are not used for chemical and safety testing, or they're not used for drug development or vaccine development. Most animals in experimentation are actually used in the category called basic research. And this is the type of research that's done in academic labs across the country, across the world. It's funded by our tax dollars, funded by the agencies like the National Institutes of Health and the Centers for Disease Control. And it can be, it's called basic research because it doesn't have to have a tangible, direct benefit for human health. So if I'm a researcher, I can, and this has happened, I can catch songbirds from the wild, bring them into the lab, scramble a part of their brains to see how that affects a part of their brains, their singing ability and their neurology. And this is the type of research that is done all the time. This is the majority of the use of animals in biomedical research. And it's curiosity-driven, just basic research. And Pratik, I'll turn it over to you if you have any other questions on that.
Aisha, hey, it's Priya. I actually do have a question about that. So when you talk about like using the songbird example that you used, so I certainly in college and in my year as a research lab tech was around animal testing, right? In college, I actually was part of labs where I performed tests on animals. And they were technically basic science studies, but it was my understanding that it always there has to be like a sort of a reason. So when you're using the songbird example that you're talking about, how is there not like a regulatory authority where they have to justify it, where it is still pure science, it's not translational, it's not clinical research, but you're still kind of tying it to human entities? Is that not true? Like, they don't have to say, well, I want to scramble the brain of this songbird because it's going to help me understand brain injury better in football players. That does not have to happen, just for clarification.
So Priya, it's an excellent question. So first of all, there's no regulatory agency that oversees this type of research. And, there are so-called bodies that either approve within each university that approves the animal research or not. But I mean, it's like the fox guarding the chicken coop. I mean, they pretty much, they're stacked with people who do animal experimentation, and it's almost exceptional for any animal research not to be approved. Now, they don't have to have a tangible end benefit for human health, this type of research. But because they've been so criticized for the suffering that they cause animals with no tangible benefit even in mind, what researchers now will do, and what they'll do in all the papers that come out on this type of research, is they'll tack on every kind of benefit they think that this research could lead to. So the songbird experiment, for example, yes, they'll, at the end of their publication in which they describe the experiment, they'll say stuff like, and this information may lead to a cure for Alzheimer's, for Parkinson's disease, for brain injury, for blah, blah, blah. And they almost always do that now for public relations reasons. But there is no legal or regulatory requirement that this research has to have some direct application for human health. The researchers will always say that, it has some kind of application for human health. But we now know how poor the information even translates to human health, and how little benefit actually humans receive from this kind of experimentation.
Thank you for clarifying that. That was my fear. A follow-up question, if you don't mind. So again, using the songbird example, or really any example. For example, we know that University of Texas has a researcher doing just incredibly cruel experiments, as they usually are, on dogs, looking at neuromuscular disorders. And these experiments have failed for decades, right? So is there a time frame where they can be told, hey, look, you've been doing this for 20 years, you're not getting any benefit out of this, so we're going to shut this down? Because my sense from being in academia is, no, they're just going to keep approving it, because there's going to be a way to go, well, no, but now we understand that this didn't work, so now we can go to this step. So my sense again is that they can do this indefinitely. They're always going to get approval. Is that correct, or is that incorrect?
It's correct and incorrect in a sense. So there is no timeline placed unless it's a stipulation of a grant, which I don't know of any grant that places that kind of stipulation, that if you don't get success within 20 years, you can't keep going down that line of investigation. So a researcher can keep doing the same thing again and again as long as he or she gets funding for it. And that's really what it comes down to. So I'll give you examples of how we have gone down the same line for decades. So if we think of HIV research, for decades now, in the National Institutes of Allergy and Infectious Diseases, actually, which is what Fauci oversees, that institute has been trying to create a vaccine that would prevent HIV for decades now. And more than 35 vaccines that have been tried in non-human primates and worked, and until the use of chimpanzees was largely disfavored, they were being tested in chimpanzees as well. Those more than 35, actually, I think it's more than 85 actually, that have been tried in non-human primates and worked and were safe, when tried in humans, all failed. And two actually seemed to increase the risk of HIV in humans. Despite the fact that they continue to fail, that institute continues to use non-human primates to try to come up with a vaccine for HIV to prevent HIV. So no, there's no, it's really up to the researcher, it's up to the funding agency, but there's no, it's up to them if they decide they want to try something else. And I'll see if Carmen, you've had some experience in this area, I think yourself, maybe you have something to add to this.
Yes. So, I think there was a period of time when I was actually involved in just kind of overseeing and doing a little bit of grant portfolio analysis for a division within the National Institute of Health. Can't really disclose which one, but we were actually looking at that. I mean, I think the federal agency was getting to a point where they really were examining if they should continue to fund those researchers that have been funded for such a long time, and maybe they were not getting what I talked about earlier today, reproducible data, in which, in fact, with animal models, you don't. Every animal, every day, every rodent is different. If you're using it, it's never the same day as the day prior. So I think they were starting to evaluate that during my time with the federal agencies, and I think they were starting to look at really carefully if they should expand out from funding the same senior scientists, so to speak, that have been in the game for 20, 30 years and really weren't coming up with any type of novel discoveries. And if they really should focus the shift more on young scientists. But again, this was more in the thought of supporting up and coming young scientists versus, really looking at senior scientists that really weren't getting anywhere. So they were trying to allocate funds elsewhere just to support the up and coming young scientists at academic centers. But that started happening during my time, and I think that as Aisha mentioned, there are no stipulation that says we will continue to fund you until you find an appropriate outcome or data to this hypothesis and thought. There's nothing there in any grant that ever says anything like that. But, the goal of the scientist is naturally to continue to try over and over and over again to reproduce the data so that they can continue to bring funding. A lot of it is that, as many know on this call, there's a lot of pressure involved with them in academic settings where they have to bring a certain amount of funding in order for promotion purposes. So, a lot of the same experimentations, to answer your question, continues, but more on the fact that they feel that internal pressure from their organizations. And federal agencies will fund them if they see that effort there. But there has been a little bit, not substantial, but a little bit of pivot towards, let's see if we can find some other young scientists that may have some better thought. So I hope that answers your question.
Yes, thank you. I have so many questions, but I don't want to be the only one asking. So I'll see if Pratik and Swati and Deep have some questions. And I know we've got Shreya on stage. And then at some point we can circle back because I do have a few more questions.
Thanks, Dr. Priya. So the way we are trying to structure this is, we have this particular question was about the reasons why animals are being used for testing. So if anyone has questions on this specific front, that's fine. Or otherwise, we can go to the next part. And the next part, of course, is going to be about funding. So does anyone have any questions about why these animals are used? Else I can just go ahead with the funding. I'll just wait for 3 seconds.
Yeah, so I actually do. So how would you guys respond to the thing that I routinely hear, right, when I hear about bleomycin induced fibrosis and things like that in the pulmonary world, in mice and in monkeys and things like that, and people will say, well, no, the reason we have to do this is because it mimics human physiology, right? So that's part one of the question. And then part two of the question circles back a little bit to the HIV model that you were talking about, Aisha. One of the things that is often said in industry when it comes to the failure of animal testing is it's never really the fact that it's the model that's being used. It's either blamed on the IP, the investigational product, or it's blamed on the fact that the study wasn't designed properly. They didn't do an appropriate interim analysis so that they could pivot if they needed to, et cetera. So how would you respond to those two questions? The first one being, how do we, what's the, eh, how do we answer this idea that it's the model and that it's not actually just the fact that, oh, that vaccine just didn't happen to work, or they structured the design incorrectly? And then the second is, how do you answer the question about animal physiology being similar to ours, and that is a justification for why animals are used?
So I will say, let me answer the second one, the animal physiology being the same. That is the sort of presumption that everyone makes, that the animal model is the closest we can to a human being. And I will say as a scientist that is flat out incorrect. There was many different times, for example, where I dissected a specific animal, and the animal had, some sort of, how can I explain it? If I was, for example, looking at the liver lobe, and I was trying to investigate liver cancer, hepatocellular carcinoma, there's a lot of times, I will say from experience, that the liver lobes of the mice from one day to the next, or to the next, mouse, was not the same mouse. Sometimes I would find that the liver lobe in one mouse would have 4 lobes, and then the next day, if I would dissect the different, same similar mouse that came from the same batch, but if I would dissect that liver would end up with 7 lobes. So there was a lot of variation, is what I'm trying to say, between the litters of the animals that actually arrived in facilities. Now, this wasn't always 100% the case, but like every human being, every animal is different, is very unique, is what I'm trying to say. And so when we're working with animals that may have some sort of differences with their anatomy, and then we're trying to extract data, consistent data from that to a human, which is a completely different anatomy from the animal, then I think it's far outreach connections. So the anatomy of animals, in a sense, to humans is not a direct correlation that we should be making in terms of trying to translate our data, because there are variations that exist amongst the litters of the animals when you get animal orders. And then to project that even further, and to connect that with, oh, so this is what we saw in the liver disease model of the animals, and now this is what we can say about humans, is completely incorrect. And that has been the argument for such a long time, is that it is the closest thing to humans because, in fact, it is a live species. But we're operating from 2 different anatomical systems that could have even variation within the same species, and then we're trying to connect it to the human. I think that thought process has to actually shift. That's not, really what's happening. What's really happening is that the data, in fact, is not reproducible. And it's because we're not using the right model. The right model would, in fact, be that we were using human tissues, human biology samples that are directly correlated to a human being. And so whenever I hear that, I just have to jump in and say that, of course, there's always that false thought that that's the correlations that people make, but it's not, in fact, correct.
Thanks, Carmen. And I'll also add, so, when we think about 100 years ago, 200 years ago, you cut open a cat and you see that, the heart has 4 chambers, pumps a certain way. And yes, you could take that and extrapolate that to human beings. There's some basic anatomical and physiological features run across all mammals and many non-mammals as well. But we're now at a point in medical research where we're looking at the subtle nuances of molecular physiology, biochemistry, neuronal chemistry, as well as genetic expression. And at this very subtle level, I mean, this is where medicine is today. We're fine-tuning drugs and vaccines and trying to understand the subtle nuances of why my twin sister could have a disease that I don't have, for example. And what is the subtle difference between the two of us that would make her have a, manifest the disease and it not manifest in me? And at this point, there's just too many unknowns and too much variability between humans and other species. And it makes you're basically playing a guessing game. I mean, the point of animal testing, why it ever started from the first place, besides basic curiosity, which was part of why it started in the first place, but the point, at least this is, what's supposed to be, was that it's supposed to inform what we will find in humans. It's supposed to inform whether a drug or vaccine is going to be safe and going to work in humans. And it fails miserably now. We know that about 90 to 95% of the drugs and vaccines that are tested in animals, that are found safe and found effective in the animals that it's tested on, when they move on to clinical trials, 90 to 95% of them fail in humans. Now think about that. I mean, that's a 90 to 95% failure rate. Most of them fail because they don't work or they are unsafe in humans. So it's not that animal testing is the only reason for that high failure rate, but it's a significant contributor to the high failure rate. Researchers will, the ones who keep wanting to cling to this old paradigm of doing animal testing will constantly say, no, the problem isn't animal testing per se. The problem is maybe how we did it or how we report the results or monitor results and so on. And they'll keep saying that and keep saying that, and they keep making little tweaks to the system and little tweaks to refine and improve reporting and improve measurements and blah, 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 but it doesn't lead to any tangible change in results. So the question we really have to ask is, how long do we keep doing this? How long do we keep doing this when there are hundreds of millions of animal lives that animals who suffer every year globally, and even more humans who are waiting for treatments, waiting for vaccines to prevent their HIV, or to lower their risk of HIV in their community. They're waiting for these treatments and they're not getting them because we continue down a very misguided, very, very misguided system of testing in the first place.
Thank you so much, both of you. Really appreciate both of those answers. I think it's so important for people to realize how flawed these models are. I mean, obviously, I know, Aisha, you're vegan. I know that there's ethical objections, but when you add to that the fact that we're failing humans by using these models, it makes it even more.
Yeah, and okay, I again have a question from Vanch in the audience, and the question is: we often hear these experiments are conducted to understand how animals react to different stimuli, or in different scenarios, and under stress, like drowning mice in water. So, what's the purpose of these kind of experiments specifically, like the ones under stress, and where you have these specific experiments?
Well, without knowing each of the particular experiments and how they, what the researchers are purportedly saying the purpose of those are, in general, they're to try to understand anxiety, for example, and fear and depression and hopelessness. I mean, I think there are more than enough people to study. We don't need to try to create these artificially in animals and then try to translate it from a very poor system in the first place, unreliable, non-predictive system, and by system I mean animal testing, to humans. That's the point. These drowning tests, for example, is to understand what they call learned helplessness. How long do you put a mouse in a vat of water, and you basically wait to see how long that mouse will continue to struggle to stay afloat before that mouse gives up and drowns. And that's called learned helplessness. And the researchers will say, oh, it will give us a better understanding of the mechanisms behind depression and so on and so on. It doesn't. Listen, I face depression. I've had depression all my life. No animal research has helped me, I will tell you that. So, we just need, there's so many people out there who have these mental illnesses, who experience these problems, who can be better served if we just turned our attention away from drowning rats and mice and instead actually focus on the people who are suffering from these diseases.
Hi, this is Shreya speaking. May I ask a quick question?
Yeah.
Thank you so much, and thank you, Aisha, for all the work that you do. I come from a science background and now work on PETA's research modernization deal and other similar ventures at PETA to help um, urge funding institutions, which I know we'll be getting into next, to really incentivize the advancement of animal-free research that's going to be more relevant to human health. So I really love that I'm able to bring my love for biology and science and my animal rights activism and really do the work that I care about. But one of the things that I do think about is this public education element that we're talking about, like as far as mental health and other important issues, where I think it's safe to say that the medications that we are taking, like I'm sure I take medication for anxiety and definitely tested on animals, but I can say that this medication is helping me. um, I wish it wasn't tested on animals, but there are medications out there that are helping people. So how can we um, be respectful of that in our messaging by saying, yes, these medications exist. They were tested on animals. They are helpful. We're not here to take these medications away from you, but rather we want to help you um, gain access to more quality medication because, hey, what works for me is probably not going to work for my sister or my people who I'm related to. So this also comes into all of that variation. So how can we, in our campaigns and demonstrations that are also part of the process, make sure that we are respectful of that in our messaging? This is Shriana.
Yeah, no, it's a great question. So, no, you have, and this is one thing that we always want to make clear. We can never, ever argue that we've never learned anything from animal testing because, of course, we have. How can we not? We've been, pouring billions of dollars into this type of work or research, and yes, maybe some medications came out specifically because of animal testing. What we do know, though, that because the FDA requires every drug or vaccine to be tested on animals, it doesn't necessarily mean that those animal tests were necessary to discover those drugs and vaccines. And so they're just part of the process. So, yes, every drug and vaccine will have been tested on animals, but it doesn't mean that they were necessary. Those animal tests were necessary to get those drugs and vaccines in from the discovery and to learn how well they will work in humans. Now, I think that the way to frame it is, it's like when, and even so, let's say there are some examples of real successes that occurred because of animal testing. The problem, if we just continue to focus on those few successes, it's like it's as though if I'm standing outside a gambling casino and I see, someone come out, every 5 days saying, oh my God, look at this, I just won, $500, or, I hit the jackpot. And what I'm not seeing are the hundreds of people still in the casino, gambling day after day after day and losing money moment after moment, spending years and basically failing again and again. And so we have to really look, when we look at the issue of animal experimentation, we really have to look at it as a whole. And not just look at maybe where there may have been some successes, but look at the overall. How many failures have there been? And if there are more failures than there are successes, and in this case, it's overwhelmingly more failures than successes, we really have to ask, is that even the right basis to continue on? And as we ask that question, we also have to ask another thing. So we now know how non-predictive animal tests are in telling us whether a drug or a vaccine is going to be safe and effective in humans. 90 to 95% of the time, they don't tell us that. That also means that we likely, or drug developers and researchers have likely abandoned, i.e., thrown away, treatments that actually could have worked in humans and would have been safe in humans. But they abandoned them because they did not work in the animals tested. So the flip side is that we likely lost out on effective treatments because of a very misleading testing regimen, which is animal testing. Does that help answer some of your questions?
Yeah, absolutely. That does. And I think that really warrants the need for more conversation. And if I may follow up really quick with one really quick question, is that okay?
Absolutely.
Oh, thank you. I don't want to take up everyone's time, but like Dr. Priya, I'm just so fascinated by the work that you do and all the questions that are really, really important to have these discussions to educate people. One of the things that I've also been thinking about quite a bit is when we have these criticisms of how animal experiments are conducted, like, hey, there's this variation, cage temperature, and who's conducting the experiment, the mood of the person who's conducting the experiment. Like, there's so many things that can influence the outcome of the data, and that's, of course, going to change day to day, lab to lab, person to person. So when we're making these criticisms, I feel like there are multiple ways this can go, and I'll pose two. One is we're making the paradigm for really high quality, consistent animal research so difficult and so inconvenient that those who do experiment on animals are going to just not want to deal with that in the first place. Or we're just going to keep trying to do this not effective thing slightly better and then reinvent the wheel and then have people going back and redoing all of these animal experiments in this more quote unquote better way. So do you foresee either of these being an outcome? How we can avoid the possibility of, just creating better thermal regulated cages or more consistent protocols? Like, how can we avoid just doing the same thing that we've been doing in a better way, so to speak, though we know that better is not really a thing here?
So I'll quickly answer that and then turn it over for Carmen, because I'm sure Carmen has some thoughts about this as well. What I would say is, that what you're talking about is basically when you think of the way animal advocates and have approached animals in research, there's this framework called the 3 R's, which is refine, reduce, and replace. And in a sense, what you're mentioning is kind of refining to some degree the testing so that they're a little bit more reproducible, having cages with the same temperature, heating in the room, and so on and things like that. But the problem with that approach is that you are still fundamentally using a flawed type of testing paradigm, which is using other species to model what we're going to find in humans. And it's a very, very flawed paradigm. So at the Center for Contemporary Sciences, we don't even discuss that. We don't talk about those things. Our approach is really to push forward a new direction in science that's towards more human relevant testing methods. And there's so many wonderful techniques that are coming out and that have come out, and they're not ideal, they're not perfect, but they continue to be improved and refined. And if we put more of our funding and more resources and more energy into these methods, they would be even better today than they were or they'll be better tomorrow than what they are today. So it really is. I would suggest, what you're suggesting, Shreya, is you're still that type of approach is still an approach based on staying with the old paradigm, and we really need to shed that old paradigm really completely and embrace a new system in which we are really using medical testing methods that are based on human biology and that are human relevant. And by doing so, we're really pushing forth into a new era of medical research. And I'll just stop there and see if Carmen has any thoughts on that.
No, I mean, I think you stated it just perfect. I think that as a scientist, that was in a laboratory space for more than 20 years, I really honestly think that the model is what's not appropriate. It's not that we need to be tweaking this part of the tested model, warming cages. I remember a lot of my colleagues sometimes would play around like feeding times. Should we be feeding the animals in the evening, when, which is when rodents eat, and maybe not do it evening and do it in the morning, daylight, that kind of stuff. Um, and none of that really worked. I mean, to shift the conditions in which the animals are being fed when they're sleeping really did not change the outcomes of the experiments. And I can say that, as a scientist that practiced for more than 2 decades. I think that what really needs to shift and alter here is the model. We need, a tested method that actually substitutes traditional animal models with non-animal systems. That's really what we need to be focusing our efforts on. And like Aisha said, that is what the Center for Contemporary Sciences is doing. We're trying to support from many different angles from and you can go to our website and find out from the educational piece of this, to policy, being involved in some of the policy decision making. So it's not refinement, it's really replacing the model. The model is faulty, when it comes down to the animals because it's not a direct correlation to the human being. And then at the same time, I will say that in your question, you brought up something important, and I'm not sure if it was in this question or the question prior, where you said, depending on the mood that the scientist is in, I do think that external factors do actually play a significant role, in terms of your experimentation outcomes as well. It's not just so much the animal tested models that is faulty to begin with, but it's also the environment that you're in. Do you have, again, the accurate, resources that needs to go with that animal model that you're using? Do you have all of that in place? Do you have the well-skilled, researchers that are well versed in animal testing methods? And a lot of them are not. A lot of them actually, in fact, get very minimal training when we talk about, these animal, tested methods, skills, research skills that they're supposed to be learning. A lot of them really don't have it. They get thrown into the research space just hoping to make magic and to assist, for those that are lab technicians. So there isn't proper training around this. There is no really good understanding of the needs of the animal. And so a lot of this too it goes back to the other aspect of this, is infringing on the rights of animals because we in fact don't understand them. We're just using them for experimentation purposes, and some folks don't even have the proper training to be around this setting. So again, I think we need to pivot, move away from, refining, reducing, and really just focus on the replacement piece. How can we investigate a human disease model? And the only answer is to use human biology testing methods, human tissues, human samples. That's the direct correlation there. And the culture of the organizations, and I'm referring to at the academic, the federal agencies and private industries eventually will get on board, and they are, but will eventually continue to get on board as they realize that that really is the best answer.
Yeah, thanks for that, Dr. Carmen. So the next question I have is, and I would actually like to keep it short, and then because we would also like to focus on the animals themselves, like the victims themselves, and Swati will take on from there. But the next question is about funding. I know, like, we talked on it a little bit, but which specific agencies, or like, okay, which specific types of agencies fund these studies, and like, is there any, like, documented motive why they want to keep using animals even when research is proving otherwise? Because I'm pretty sure they do know, like, about the fact that we have such a high failure rate. So do we have like any specific answer for that? But like I said, I would like to keep this part a little short. Over to you.
Yeah, I can take that. So, the Food and Drug Administration funds animal testing and conducts animal testing. The Centers for Disease Control does that. The National Institutes of Health does that. Our military does that. Department of Homeland Security does it. U.S. Department of Agriculture does it. And then, of course, there are lots of, hundreds, if not thousands, of medical charities that fund and or conduct animal testing, like the Michael J. Fox Parkinson's Foundation. And I think the reason why they, even in light of the failure rate, first of all, many actually don't know about the failure rate in animal testing, because many researchers, and even when I was at the FDA, many people within the FDA don't even know about that high failure rate. They're kind of in their silo and they work in their specific area, and that's where they stay focused, and they don't think about or are aware of the larger concerns. And I think part of the reason why despite the incredible high failure rates, is that there is truly it's been ingrained culturally, this misinformation that we need animal testing to better human health. And so it's a hard culture to change, but I think that we are going to start to see that cultural change happen with younger scientists, the up and coming scientists who are going to be approaching these issues differently. I mean, the Food and Drug Administration, NIH, all of them, they're packed with career scientists who they did animal testing, they know animal testing, that's what they're comfortable with, and that's what they continue to ask for.
Oh, thanks, Dr. Akhtar. And I'm actually surprised to find, find USDA in that list. But yeah, over to you, Dr. P.
Oh, no, I think Swati had one, and then I'll go after Swati.
Hey, Swati, did you have any question?
Yeah, thank you. So, yeah, I just want to bring the focus back to non-human animals as such. So, of course, we know whatever happens to non-human animals is non-consensual. Of course, it is. But the treatment of non-human animals in animal testing, right from breeding those non-human animals into existence, can you shed some light on that? Over to you, Dr. Kadir.
Sure. I'm trying to understand your question. I'm sorry. Can you repeat it again?
Sure. So Sure.
Go ahead.
Sure. So the question was about the treatment of non-human animals in animal testing. So, of course, right from the breeding part, right, these non-human people are bred into existence in these facilities where they are there's batches of these non-human animals used for these tests. So, what is the living conditions of these animals, and like the procedures that are conducted on them? What is the standard of life for them?
Sure. It's a very sad question to ask, actually. I mean, a very important question, really sad question at the same time. When I think about it, because I spent so many years doing this work, I guess, all the facilities across the country, and I can only speak for the United States, are very different. But I will say that, they do have, they're supposed to have like a vivarium, like animal team that is there during the daytime hours, that is, signing the shipments of the animals when they come in, and they do get transported in cages. Um, a lot of times there's more than one animal in cages, especially with rodents. You'll find that sometimes there's, litters packed in a cage, which is a very sad sight to see. And then, the vivarium crew, which is those staff members that are in charge of overseeing the animals for research purposes, then start separating the animals into their own cages. And sometimes, they'll have no more than two, is what I've seen in my time. And I think that, they definitely, depending on what that animal is going to use for, is what that animal will be used for. There's sort of like a diet that gets assigned. So each animal in each cage will get a specific diet. protocol that will get assigned. Some, which is, when you think about this, it's crazy, right? Some animals have diet restrictions. They can only eat specific foods because of whatever underlying medical research the scientist is going to perform. Some animals have to be on for example, for those investigating kidney disease in the Department of Nephrology, a lot of times, they're studying hypertension, right? Which is high blood pressure. And a lot of them will have these animals on a salt-free diet. So these animals can't have any salt intake, which totally minimizes, again, like what they can eat and not eat. So again, their conditions and their reality is based on what they're going to get used for, what their medical experimentation purposes will be for. And that's what their diets are assigned. So again, they arrived in batches where they're a lot of them in one cage. It's not until they get to their destination that they get separated out. And then they're put on these diet restrictions. Sometimes they're put on specific light restrictions, lighting, photosensitivity, depending on, again, what other underlying mechanisms the scientist or physician scientist is investigating. So there's a lot of restrictions based on what the animals are going to be used for. And that's the piece where I think, is not correct. That's the piece where I think we are infringing on their rights. That's the piece where I think we're not being compassionate scientists. And so we are creating an environment for them that is not a natural environment in their existence for our own research purposes, where we will, instead of creating a disease to cure it, we're creating symptoms of the disease and then reverse those symptoms. And that's really the reality that these animals are in until the day that they get put to sleep.
And
Doctor,
thank you, Carmen. I can also add a little bit about some of the regulations. So in the United States, there's the Animal Welfare Act, and it's the only federal regulation or federal law that has any impingement on the use of animals in experimentation. Now, it's really important to note, though, that the Animal Welfare Act excludes the majority of animals used in experimentation. It specifically excludes mice and rats. They are not defined as animals under the Animal Welfare Act. It excludes birds. It excludes all non-mammals and invertebrates. And it excludes any animals used in agricultural experimentation. So the Animal Welfare Act doesn't even cover the majority of animals used in experimentation. And because of that, we don't really know how many animals are used in the United States because they're not counted. Mice and rats and birds and reptiles and all those other critters are not counted. Now, for those species which are covered under the Animal Welfare Act, it's pretty dismal. It provides a kind of a checklist approach. So if I'm running a laboratory and I have, a laboratory of dogs and non-human primates and cats, for example, under the Animal Welfare Act, I have to count how many animals there are in the lab, and then I also have to say, check off like, yes, I did provide food for, I do provide food for the dogs, or I do provide dogs a little room to run every day, or the cages have to be a certain size for non-human primates. The important thing to note about this, though, is even for those animals which are covered under the Animal Welfare Act, under no circumstances does the Act provide any limit on the amount of suffering and pain an animal may experience in the name of research. Meaning, if I want to burn and radiate a pig alive, if I want to say I'm not going to give that pig pain medication because it will interfere with the study, I can do that. The Animal Welfare Act does not prohibit me from burning a pig alive and from not giving that pig pain medication to ease her suffering. If I'm running some experiments on dogs, and even though the Animal Welfare Act says I have to feed them, if though feeding my 12 or 50 dogs is going to interfere with my study outcome, I don't have to feed those dogs. I can starve them. So the Animal Welfare Act provides no limits, provides no restrictions on what can be done to animals. So thousands, there's I think about 60,000 animals of those species covered under the Animal Welfare Act. Remember, that's less than 5%. But of those less than 5%, in 2018, 60,000 of those animals were used in experiments for which that were considered painful, but for which they got no pain alleviation whatsoever. And that can happen because of it's really because it's such a pitiful regulation. And I'll just stop there.
Thanks, Dr. Aisha. I think Deep had a question, and after that, I have like a short follow-up, and then Dr. Priya.
Dr. Carmen, yeah, pardon me if this is a silly question, but would you say to use the term compassionate scientist is an oxymoron? Because I mean, you can either be compassionate or a scientist. Have you come across stories where scientists have actually stopped seeing these animals as commodities and had some empathy for them and became whistleblowers at some point? Does it take us, I'm sure that it was a gradual process where they reached a point where they could be insensitive to the suffering of these animals, but then would you say some part of the brain or that empathizes like normal humans do has been switched off? You get what I'm saying?
Yes, I do get what you're saying. I think you said it actually very briefly in your question. I think that it is a very slow process. It's a slow transition, to be honest with you, I'm an example, where I worked in the laboratory space for 20 years, and for so many years, I knew two things that I did know. One, as a professional, I knew that the data, right? When you talk about scientific data, and you guys hear about this through COVID-19 now with the vaccinations, right? What does the data show us? Real-time data. Are the vaccines being effective or not? Well, every scientist, that's the main question on their mind is the data. What is the data telling us? How can it guide us to the next sort of discovery phase for us to investigate this disease? And I knew that the data was not being reproducible. I knew that the data was in fact very sloppy. And I knew that that was the first realization for me. We're not really capturing what we need to capture. As we continue to go up to the 11th floor of that vivarium and continue to do medical experimentation and sacrifice these animals, we're still not getting the answers. Simple answers that could perhaps be seen in other testing methods. Like when I would use human biopsy tissues, I would actually, in fact, get more consistent data than I would in the animal. So that was my first realization. And then the second, which really is the first one, the most important realization to all of this really was that I was imposing my views as a scientist and what my thoughts were in the hypothesis that I had created and formulated to investigate diseases. And I was violating the rights of other animals. I was not really being respectful of their existence. And not only was I doing that by putting them in restriction diets and restricting how much light they could get in the daytime or when they could sleep and not sleep, repetitive experimentation on the same animal. So where you would suture the animal back up, the way, you see surgeons doing in the operating room. They suture patients and the patient survives. We would suture animals back up under anesthesia. We would put them to sleep, suture them, and we would put them back in the cage to heal. We would give them time for healing, and then we would reuse the same animal for experimentation purposes to then get more faulty data. And the suffering of the animal in the process was, to be very honest with you, very unbearable. I thought it was just some of the very saddest moments of my career to see these poor animals suffer through continuous experimentations on their bodies for faulty data. There was that season of sort of reckoning I had to go through where I knew this was not correct. Not only was I not enhancing human health outcomes, because I wasn't, I was also infringing on the rights that these animals have. And what ultimately does happen, I will say this, is that you do become more compassionate. You do become more respectful towards other life, towards other beings. And one of the programs that we're creating, and I'm just going to say this really briefly, at the Center for Contemporary Sciences, is educational programs where we will in fact be teaching 21st century scientists how to be more compassionate, how to be compassionate scientists in the 21st century. And what does that mean? It means exposing them to what human relevant tests and methods are. What are some of these kits out there, these organs on the chip? What are some of these other advanced technologies that we now have in the 21st century that we could be utilizing to not only enhance human health outcomes, but to also protect and be more compassionate towards animals that exist around us, that have the rights just as much as we do? And I think that in our educational program, our hope is that we can enlighten students across the country to actually have them recognize that there's other ways for them to conduct compassionate biomedical research science. And it also will create not only a more compassionate scientific community, but also I think it will help create more workforce retention where scientists are not quitting, for example, where they feel disenfranchised like I have for 20 plus years. And I left the basic science field. I left because I couldn't continue to get faulty data and not improve health outcomes for humans. But I was also violating animals by continuing to perform experiments on the same animal over and over again. Harming them and really just not being compassionate. And so our goal at CCS again is to create this more compassionate scientific community and to have these students, young budding scientists, understand that you can exist in the scientific space, you can continue to conduct research to investigate these complex diseases. You can continue to test drugs and toxicities, but on human biology / tissue samples where you could get more consistent data and you could improve health outcomes and you're not really interfering or interjecting your right on animals.
Yeah, thanks for that. Thanks for that, Dr. Carmen. And before I move ahead, I actually want to first applaud you and your team for just for saying that okay, I have seen this and I don't want this to happen at least on my watch. So that's really applaudable. And I would also like to empathize with everyone working in these fields to see these animals suffer. Of course, it's much more worse for the animals, but even for humans working in these fields, it's definitely a traumatizing experience. So thanks for trying to move the needle. So my question was again, so for example, countries like the UK, at least, and I don't know about US and the other parts of the world, they do give sentient status to the animals. Now that definitely does not change anything for the animals because we are still killing the animals, at least in other fields. But then you have regulations where if you have the sentient status, then you can't subject the animals, for example, in food industries, to be boiled alive. So does is this field being excluded? I mean, if the animal still has a sentient status, uh, can you still use the animals and make them suffer or make them starve? I don't know if you have any information about what happens in the UK, but
Oh, Carmen, go ahead if you want to answer that.
No, go ahead. I actually don't know what the protocols are in the UK. That was actually my answer, but
Okay, sure. And so, yeah, first of all, I just want to say, it's rare to hear from someone like Carmen who has gone through that journey and, to be honest about what she experienced. So thank you so much, Carmen, for sharing that with everyone. And as far as the sentience, unfortunately it doesn't really have any role currently in animals used in experimentation. These animals. We know dogs are sentient. We know cats are sentient. We know mice and rats are sentient. We don't even need that definition. I mean, the whole point why rats and mice are used in like depression research is because they can experience depression, is because they have emotions, is because they feel fear, is because they experience pain, is because they experience anxiety. And so, it's been well recognized for a long time that animals are sentient, and that's why they're used. But because of that, I would say that it hasn't gone to the point where we actually start thinking about, wait a minute, if rats can experience depression, doesn't that mean that we should really rethink how we use rats, and rethink the use of them in these experiments in the first place? And that kind of conversation, that hasn't really happened, not to the degree that it should. So it's still kind of this false narrative when it comes to animal testing that still is propagated, meaning that there's a narrative that the public still largely believes, policymakers still largely believe, and even scientists, they're trained, they're in, and through the scientific culture, they're kind of educated to believe that we have to do animal testing to protect ourselves. And so that desire to protect human health overrides any other concern about animals and their suffering. And the truth is that actually we have to move away from animal testing to protect ourselves. That it's a win-win situation. Moving away from animal testing, embracing more advanced techniques, human biology-based techniques that could be so much better in telling us what we're going to find in humans, that can more accurately replicate human physiological or be better human physiological models than any animal, any other species could be. This is actually, what's better for humans and animals. It truly is a win-win approach, and this is how, I think the narrative needs to change to show, no, actually, we're better off, and we can protect animals better by moving away from animal testing.
Thank you,
Okay,
Aisha.
thanks a lot.
Yeah, so I do want to say one thing. I think we should be careful about framing scientists as not being compassionate. I think, to Carmen's point, to Aisha's point, I've certainly experienced it myself. I've done things to animals because I was taught that it was a necessary evil. Everyone acknowledged that nobody really wants to be doing experimenting on animals, but I was brainwashed into believing it was necessary, right? Doing procedures on pigs in my training was a necessary thing in order for me to be a good doctor, and of course, none of that was true. So I just want to be clear that that most scientists truly are compassionate. We just need to start extending that compassion to non-human persons, and in such a way that we can be like Carmen, right? That we can break away from that and say, no, I'm not going to do this anymore, and that we can stand up for
So,
non-persons.
so yeah, so thank you for bringing that point up. So that actually is what we were referring to. Scientists are very compassionate individuals in the sense that they really truly are in this lab space, working ridiculous hours, not just during the week, but on the weekends. They don't have a set, let me go in at 9, let me get off at 5. There is no 9 to 5. Their commitment to the work is incredible. And then a lot of times their schedules are actually dictated by the experiment. The experiment tells them how long they have to be in the lab, and if they have to come in on weekends, on holidays or not. You may be running an experiment where it requires 5 consecutive days, and maybe you started it, days that overlap into a Sunday, days that overlap into a holiday event. So very committed individuals, very compassionate indeed when it comes down to the fact that they're trying to extend human longevity. That's their purpose. When we talk about, now I'm referring to biomedical scientists, they're they're there to investigate these complex diseases. They want to know what what is this underlying mechanism that is involved? What is this cell signaling pathway that's being triggered that is causing these tumors in different parts of the body, that is causing these neurological deficits, that is causing some of these muscular skeletal weaknesses. So they're there to investigate the most complex human disease questions of all our time, and to come up with answers so that they can extend my life, your life, and everyone else's life. And then what their work is on the back end obviously fuels the physicians on the front end to how to treat. How can they better treat patients at the bedside? So definitely full of compassion, full of heart to come up with the right answers to very complex illnesses that has taken people's lives sometimes very too early. And so I definitely agree with you, and I thank you for making that point. They're extremely compassionate professionals, but when it comes down to, the compassion that they're blind, the lack of compassion because they are blinded when it comes down to working with other species, that's the part where I think it's lacking. And a lot of it is because of the culture of the organizations. The culture of the scientific community has been the only way you're going to get a drug discovered and developed and approved is if you test it in a live creature. And that, in fact, and I know this as a scientist, is not true. That is not the only way you're going to get that answer. But again, the culture of the organizations have, like you said, brainwashed. They've instilled these thoughts into scientists to think that this is what they have to do to remain in the research space and to be successful in their careers. And it's in fact not true. And what we're doing by default with us not really thinking about it is we're indirectly violating the rights of animals. We're not thinking about it. It's not like there's this awareness. At some point there is an awareness and we can choose which way to go. The way I've chosen to decide this is not correct. Not only am I not getting reproducible data to extend your life, my life, and everyone else's when it came down to specific diseases that I was investigating, but at the same time, it wasn't right to see the suffering in these animals over, and especially the ones that we were using for more than one experiment, where we would anesthetize them and then we would put them to sleep, allow them to heal, and then do it all over again days later. That was the part where the compassion by just that robotic motion that we were in was not there. And so at CCS, what we're trying to do is create that self-awareness. Are you really paying attention to what you're doing in the research space? Are you in fact getting the outcomes that you became a scientist to get? And by default, the models that you are using, is that at any point, shape, in any shape or form, are you, violating the rights of those creatures that are voiceless and are not necessarily the right thing to do to be multiple experimenting on them to get data you're not getting? So we're just trying to bring that self-awareness by introducing these other advanced technologies and tools that are out there in the market.
Oh yeah, so I just want to be clear. That wasn't a critique of anything you had said or Aisha.
No, I know. I know.
That as vegans, I think we have a tendency to demonize people who are still in these fields, and we sort of
Sure.
fact that they are human, and as you've said, and as I personally experienced, many of the people that I worked with when I was doing Huntington's research, when I was doing research on Gulf War syndrome, the days, like you said, the days that they did what we called sacking, right? Sacrificing the animal, which the language in and of itself is complete crap, right? It's not a sacrifice to us, right? We're taking the life of another being, right? It's one of the saddest days for most researchers, and we continue to plow ahead because we've been convinced by the system that this is what needs to be done and it's the only way to do it. So that was really the point. But you've actually brought up something that leads to my other questions, which are kind of related. You talked about some of the diseases that we're looking at and that doctors are trying to work on. One of the critiques I get when I talk about organs on chip, right, lung technology, things like that, and as a pulmonologist, the lung on chip in particular is very interesting to me. So one of the critiques that I often get is, well, it's twofold. One is, well, the only way to validate that system, right? Because everything in medicine we try to validate, right? If you're going to make something the new gold standard, you want to compare it to the existing gold standard. Is that people still believe that we have to compare it to animal models in order to validate it, which seems inherently flawed, right? If you're comparing your model to an inherently flawed model, you're not really proving that your model is a good model. So that's question number one. How do we answer people who have that criticism? And then the second question is, the other criticism that I get is, if I want to look at, say, lung fibrosis in a lung on a chip technology, that's an isolated organ on a chip. And people are always going to say to me, well, but you have to look at the whole animal, right? You have to look at the whole model because the immune system is involved and all these other isolated systems are involved. So how do you answer that criticism of, for example, organ on chip technology, that you're looking at one isolated system and you're not looking at how the lungs might interact with the immune system, or might interact with the kidneys, or might interact with the heart?
So, Priya, let me just start with the second question. So, yeah, animals are whole systems, but the problem is they're the wrong systems. And actually, you're adding more confounding variables when you're looking at multiple interactions that we don't even know about what's going on in another species. I mean, we know still so little about the neurochemistry of each particular species, and the biochemistry, and how the genes are expressed, and play in the differences between not only a mouse and a rat, but between different strains of rats and different strains of mice. So they're the wrong systems in the first place. The second part is, when people say we don't know if they're validated and we need to validate these tests against animals, you have to fall back and turn back to them and say, what is the ultimate goal of this research? Is it to find a drug for pulmonary problems in mice, or is it to find a drug for pulmonary problems in humans? Which of course is the latter. And if that's the case, then we need to go back and validate the testing method against human biology. And that's always what we need to do. And yeah, it doesn't make sense. When I was at the Food and Drug Administration, my office actually gave a several $million grant to one of the institutions to create pulmonary or lung organ on a chip technology, because we knew that the animal tests were incredibly misleading. I mean, that was the whole point of funding the organ on a chip technology, to do safety studies that could better predict what we're going to find, how a drug or vaccine could affect human lung tissues. But then the FDA turned around and said, oh, but you've got to validate this against the animals. And it just doesn't make sense. We're doing it because the animal tests were wrong in the first place. That's why they were funding organ on a chip technology. So they're just kind of, it's like they really do. So many people still work with incredible blinders. It's like, it's like viewing the world through animal testing tinted glasses. They cannot see outside of animal testing. Animal testing is what they know. Animal testing is what they keep asking for, even if it doesn't make logical sense. And then the other thing I will say is the fact that organ on a chip technology, for example, is relatively novel, and like I said before, it's not perfect. There still needs improvement, and there's many, and it's not going to take one type of testing method. That's, first of all, it's going to give us all the answers we need. But the fact that they are not perfect is used to justify continue putting money into animal testing, and that doesn't make sense, because those methods are not as good as they could be, because we continue to divert money that could instead be used to better organ on a chip technology, or to discover better methods of testing. So and then the last thing I will say is that actually organ on a chip technology are now being used to replicate whole body systems now, because, for example, there's a lung on a chip, there's a human heart on a chip, there's a kidney on a chip, a gut on a chip, there's a human mini brain on a chip, there's a nerve vessel on a chip, and so on. And they're actually being connected. So you can actually see how a disease may run its course through the whole body, or how a drug may impact multiple organs and how they interplay with each other. So these advancements are being made. They can be made, and they can be further made if we put more resources, and that really means taking resources away from the repeated failures that we've been having again and again with animal testing, and instead putting them into improving these other more advanced testing methods that are based on human biology, and in discovering what is the next big breakthrough in medical research going to be. What's the next thing that's going to be even better than organ on a chip technology?
Thank you. So I mean, obviously I agree with everything you said. I'm really hoping people will start looking at ex vivo technologies. We use them in lung transplant all the time, and I'd really love to see that grow for other organ systems as well. We put kidneys on pump all the
I have a quick question about the going back to the ethics that we were addressing.
Yeah, sure, Shreya. And if you can, like, please keep it like a little brief, because I think we are also running out of time. And maybe like even Swati has like one or two questions. But before that, if anyone else has any questions, you can please like back channel us the questions, and we can reach out to Dr. Akhtar, Dr. Carmen, and anyone in their team, and probably also like see if we can have like another session like in the new year or something. But yeah.
Sure, yeah, I'll keep it brief. So I personally haven't worked on, well, I did work on worms in undergrad, but my goal getting into research was to replace the use of animals using these in vitro organoids. So I've worked on intestine and I've worked on kidney, and it's really fascinating the development that's happening in that area. But one thing that I hear from other students is that outside the desensitization, I find it really interesting how they try to rationalize the dissonance that they're feeling. And one argument that I did, not really argument, but kind of a way to appease oneself, was that I make, and this is what another lab tech had told me, was I don't like to have to kill mice, but when I think of it as they were bred for this purpose, and they have fulfills their purpose, and so that makes me feel better. How does one necessarily respond to that in a way to encourage them to not lose that grip on that the fact that they know that what they're doing isn't, it doesn't reflect the morals and beliefs? This is Shriana.
I guess the question is, why does the fact that they may be bred for something have anything to do with how you're supposed to treat them? I mean, I guess some people could say that, in certain countries of the world, children are groomed to be child brides. And does that make it right? Does that make it, does that mean that you are, that they're meant to be that, and so on. So I've heard that too, and I guess it just goes back to really turning it back on them and maybe framing it in a different way. One thing I always find is when you frame a question, you take a human scenario that's very applicable or very comparative, and you frame it back to them. I find that often then they start to really think about it in a different way.
Yeah, thank you.
Yeah, thanks. Hey, Swati, I think we are like close to kind of wrapping up, but I think we have like two very important short questions to ask. So, Swati,
And
if you
Pratik,
want.
we should also make sure because Mahesh has been sitting up here for a while, so we should make sure he gets an opportunity to ask us.
Oh, sure. No worries. Okay, fine. We'll get to Mahesh first, and then we can ask Swati to go for her questions.
Yeah, it's a quick question. So it's about plant-based vaccines. It is my responsibility to elaborate my question a little bit here. Everybody should take their vaccines. So I've heard on many platforms that because of this plant-based vaccines that has been used as a as a tool for anti-vaxxers campaigns, but it is entirely not so. We should take the vaccines as soon as possible. That is the only way to get COVID out of this world. So now coming to my question, it is a completely scientific one. So do not judge this based on any personal biases or political or any other things. That is a disclaimer I have to make. So my question is, uh, I have read this article, and I also have gone across a couple of research papers that says about plant-based vaccines for different diseases. So, does developing a plant-based vaccine reduce animal testing in any ways? Thank
Carmen, if you want, I can take this one. Okay, yeah. So yeah, I've been reading some things about plant-based vaccine. They're just vaccines that are been with ingredients from plants. I mean, but the testing regimen's going to be the same. They still have to be tested per FDA's regulations. They're still going to undergo animal testing like any other vaccine. So it doesn't change anything in regards to the animals that are tested on, how many, and the replacement of animal testing.
And a follow-up question: the influenza vaccine. I've heard that it is produced by using eggs. Does this technology offer anything from? I mean, does it reduce the use of eggs and chickens from being used?
Well, there's influenza vaccines now that you can get that are not. It's becoming now the new slowly. I don't know if it's completely overtaken yet, but that they're cell based, so they're not being used. Developed using eggs. So yeah, there's already influenza vaccines. Every year you have the option of getting the non-egg based, and I think that's going to be taking more and more of a priority. But again, these vaccines, these kinds of influenza vaccines, unless it's for a completely novel influenza strain, they don't require animal testing. I mean, because they're just variations of the batches that we've had before. So they're pretty much just routine now, and many of them are being made without using eggs.
Thank you, Aisha. Thank you for that. And one final question, quick question. So I've read this on this paper that when we make influenza vaccine on an egg, there is high probability that the the vaccines can, when we I'm not from a medical profession, I mean field. That's why I'm getting stuck in between my words. So please forgive me for that. So when we use a egg, there is high probability that this get interacted with the egg, genes in the egg, and that can become less effective in when it's applied to a human. But whereas when we use it on the plant based one, there is higher efficacy. Is that true?
I'm going to have to tell you I'm ignorant on this one here. I don't have an answer for you.
Yeah, I think I
Thank you,
think this
Aisha.
is kind of
I'll just
not a,
back channel
yeah, yeah.
that research paper I read. So yeah, thank you. I'm so just for the audience, this is not something that we are sure of. So do not take it as an
As an
authoritative statement.
authoritative, yeah, okay. Yeah, thanks a lot, Mahesh, for that. And so Swati, do you want to just go ahead with your questions? And I would also like to ask Dr. Aisha if she can check her back channel because I have a message.
Yeah, sure. So, I just want to talk about the new bill is coming up, right? The Fda Modernization Act of 2021. So with this new bill coming up, what is the scope of other human relevant testing methods? And is it like a pivotal bill, or is it just like the same things we see, right? Raising animal welfare standards, and just like a what can I say? Just like a just on show, or whether it is actually going to bring some revolution in it. So yeah.
So yeah, right. The FDA Modernization Act is the new bill. It's the FDA Modernization Act of 2021. It was introduced into the Senate in October. Senators Cory Booker and Rand Paul and other Democrats and Republicans are co-sponsoring it. Basically, what it does, it goes back to that original authority that Carmen mentioned at the beginning of this chat, that 1938 statute that pretty much set up the FDA. And in the statute, it stated that all new drugs have to be tested on animals. Well, that's a Depression era statute. You know, science has changed. It's improved since then. And so what this new bill, the FDA Modernization Act, does is it just goes back and expands the type of testing methods to include other more human relevant testing methods. So it would give, if it were passed, it would give a drug developer different ways of testing that could replace the use of animals. So it's a first step, a necessary step, and if it passes, it could have a real role in moving the marker and moving the drug development and testing paradigm further and further away from animal testing and more towards more of these human relevant testing methods like organ on a chip technology.
Thank you for sharing that. And before I go ahead, Prateek, do you want to ask?
I think to
No,
make sure that Carmen and Aisha have time. I know that we had slated 90 minutes, but you guys are just, you're educating us so much. We also want to make sure that you have time to give us all sort of a breakdown of what CCS is, what you guys do, how we can help, what else we can do, whether it's calling Congress, things like that, to sort of help usher in a better era. Do you all have at least 5 or 10 more minutes? I know that we've already gone 10 minutes over.
Sure.
Yeah, and why don't I just let Carmen maybe start to wrap up by talking about some of the programs that she's working on at CCS.
Could we
Sure. Go ahead.
general what CCS is, because it's it's a nonprofit is my understanding, right? So you guys accept donations, things like that.
We do, definitely. We're a nonprofit organization, and our role is we don't create the technologies like the organ on a chip technology. What we're doing is we're creating an ecosystem that supports moving away from animal testing and moving towards better methods. So we work by changing policies like the FDA Modernization Act to allow more human relevant testing methods to be used in drug development. We work with educating scientists, educating up and coming scientists. Sometimes we help match investors with companies that are creating these non-animal, more human relevant testing methods. We really are working hard on trying to change the narrative that it's not us or them, basically. It's not us humans versus animals. It really is humans and animals. It's a win-win by moving away from animal testing. So that's kind of a brief overview. And then I'll let Carmen talk a little bit about some of her specific programs, because there's one in particular that's coming up in.
Sure. So thank you. The programs that I'm focusing on, the first one is what I'm just going to say really briefly because I already talked about it, was the academic outreach piece where we're trying to foster the next generation of scientists. And we're doing this by working with academic institutions to build curriculum and human biology focused research. So we hopefully will be able to do that with academic institutions in addition to offering our coursework around human relevant testing methods onto virtual learning platforms. So that should be rolling out in 2022, and I'm leading that effort. The second one, which is really exciting for us, is we're having our very first ever investor summit. CCS is leading this, and what we're doing is we're essentially just driving that support for companies that either are in the startup phase or would like to scale. It could be companies or institutions, and sort of connecting them with potential investors that are interested in supporting their businesses and to continue to push the needle even further with these human relevant tested methods. We have knowledge that the market in this space is going to continue to grow. We know that forecasting here in the next 10 years, even though animal tested methods continues to rise, human relevant tested methods will continue to rise as well. And even further than it has been. So the market is definitely going to be there in this next decade that we're talking about, from 2020 to 2030. And I think that it's important for us to be able to support some of these, potential businesses, companies that are trying to either start or scale by connecting them with potential investors to do that with. And, again, these companies are really trying to create technologies that will replace animal testing and eventually will have a lower carbon footprint. So we're very excited to be a part of this. And this investor summit is actually coming up in February 23rd of 2022, and we're doing it in partnership with Beyond Animal. So if anyone is interested, please check out Beyond Animal's website and register to attend this event.
Thank you. So we've got the link for Center for Contemporary Sciences up at the top. So for anyone who wants to find out more information, please go to that website. And again, I can't emphasize enough, it is a nonprofit. So donations obviously will help the organization, which ultimately helps non-human persons, which is hopefully the goal of everyone in the room. Anything else that you guys have going on or that you'd like us to know about? One question that I would have is, in addition to working with CCS and helping you guys, are there other things that we can be doing? For example, what is the status of the Fda Modernization Act? Is this something that we should be calling our own representatives about? Are there other things that we can be doing? Are there other organizations that you like? For example, I know there's the Center for Alternatives to Animal Testing at Johns Hopkins.
So I would think that one of my favorite is in Canada. So if there's anyone here in Canada, there's the I always blank on the name. The center. There's the
Canadian Center for Advancement something.
Yes, is Charu. Her center. She's just a wonderful person, fully dedicated and smart. So hers is an organization I really like. As far as what people can do, yes, the Fda Modernization Act. There's actually a pretty decent chance. It's hard, as you can imagine. First of all, getting a bill even into the Senate, and then even harder to see any bill actually pass, especially when it concerns animals in any way. But we have optimism that this one could actually go through and get passed. And so, yes, please. I don't know what the websites are, but if you just Google like how to contact your representative, it's in the House and in the Senate. So you can find out who your representative is in the House and Senate. You Google, there are links that give you the exact information you need. All you have to do is say, I'm calling to say I support the Fda Modernization Act of 2021. They don't care about anything else. They don't care about why you support it. They don't care about, what your background is, none of that. All they want to do is count how many people are for and how many people are against. So that's all you need to do. Or you can send an email. And then, please, join us on our Facebook, on our social media sites. You can subscribe to our newsletter, and you will hear about more opportunities that arise, how you can help, and maybe if you are a scientist in the biomedical field, if there's something that you can more directly do, you will learn about these options as well, and hear about any updates like the Investor Summit that's coming up, or We're going to be hosting a series of webinars in 2022. You will learn about what those webinars are, and they'll all be free, and just sign up. So please do join us either through social media or sign up for our newsletter, and we'll keep you up to date on any upcoming activities.
If you do have time for just one more, do you see a future where, instead of medicines being mass produced, it is, medicines are individual specific, which takes into account the comorbidities of the and the physiology of the patient. Like sometime in future, I know that we are like a very long way away from that. Is that even a possibility? And if we need to get there, then do we need to switch to human models as soon as possible?
Yes, the answer is yes to both. So Carmen can also talk quickly about this too. It's just we're talking about the era of personalized medicine, in which drugs and are tailored towards each individual's unique biology. So we know that just because two women may have the same form of breast cancer, for example, doesn't mean that the drug is going to work the same. It may work in one woman but not the other, or it may be safe in one but not the other. So we will be embarking on an era where we can actually test and screen for medications and test to see if they work with your biology and against your diseases. And in that role, animal testing really doesn't play a role, because we're really talking about, for that to embark in that era, we're talking about the need for testing methods that are based on human biology and that capture the diversity within the human species. And so that really is where we're headed, and animal testing will have I predict it's because that's the area where we're headed, that means that we're going to be moving away from animal testing and more towards capturing and really understanding and studying the variations in human biology.
Hey, yeah. So thanks a lot again, Dr. Aisha. And I know we have already crossed like the time limit, and I'm thankful that you agreed to stay over time. So I will like just let you and Dr. Carmen conclude the session, but I just want to stress on the fact, and like for the audience, please do go to the website that we have pinned up here, the Center of Contemporary Sciences. Sign up to the newsletters. Um, Donate if you can, because that's the only thing that we can do something. People who are not actually working in this field, that's the only way that we can support these wonderful people. And of course, like Dr. Aisha said, just call up your representatives and ask and tell them that we support the Fda Modernization Act. So yeah, Dr. Aisha and Dr. Carmen, if you want to add something, if you want to say something, please go ahead. And for anyone else, if you have any more questions, we can definitely email them and like ask the center for answers if they have any. So yeah,
Well,
Dr. Priya, you want to say something? Otherwise,
ask, and no pressure here whatsoever. Just think about it. Maybe we could do another room in the new year if you guys have some time, because it's such a complex issue that I think it's hard to answer all of them in any one setting. And certainly, thank you for letting us know about the webinars. That's something that I've really enjoyed your webinars in the past. So I would really encourage anyone who hasn't had an opportunity to be involved to do so. And Aisha, anytime I can do anything, I'll send you an email. I'm happy to contribute in any way that I can.
I know, Priya, you're just wonderful. Thank you so much. And yeah, we're definitely up for doing this again. I don't think I really have anything to add, except just, I've been working on this issue for more than 30 years. And I will say that for the first time, I actually have optimism that we can end animal testing in our lifetime. It's never soon enough for me, but I actually do have optimism that this is one industry that we can end. And it's going to end mostly because of the increasing, growing recognition, even within the scientific community. It's happening. It's not at the tipping point, but it's happening. The increasing recognition that animal testing has basically, we've reached the end of what we can do as far as extrapolating information from other animals to humans, and that we need better methods that are human biology based. So that gives me a great deal of optimism. And then I'll turn it over to Carmen, see if Carmen has any last words.
No, just wanted to thank you all for this opportunity to, discuss this special issue and this topic with you all here today. Just really echoing what Aisha said, I really think that we're at the tipping point where if we really are going to start focusing on what personalized medicine and precision medicine is, we really need to pivot more towards models that mimic human beings and really try to match the biology, so to speak. This mismatch in biology is in fact causing many useless and just toxic drugs to get it through clinical trials. And then potentially also, we're hindering the opportunity for effective compounds to never really even get discovered, because some drugs we're seeing aren't working in animals, and then that's the high failure rate. And then some drugs that, we're also missing that even if they're not working in animals, they could have worked in humans. And so we don't know. And I think that we're at that pivoting point where we really need to focus on how we can have better discoveries in a similar, matching biological system. So really happy to be here. Thank you so much for including me in this conversation. And, I definitely look forward to having future conversations with you all. Thank you.
Thank you guys so much for having us.
Thank you guys. Follow both Dr. Carmen, Dr. Aisha. Follow the club, follow anybody on here that you connect with. Look to your right and left in the audience as well. And then, yes, please, please, please call your representatives and your senators to say that you support the Fda Modernization Act of 20.
Thank you all so much.
Oh, sorry, go ahead.
Oh, that was it. I was just saying thank you very much for having us.
Yeah, I would just let Swati end the discussion. But before that, I specifically want to thank you, of course, the center, and Dr. Priya for putting us in touch with you, and Swati for being very proactive in reaching out and setting up this whole thing. And everyone in the Planet Earthlings team, they've worked a lot and they do work a lot. And also everyone in the Global Jain Network. I know Sunny is in the audience, and they, like the Global Jain Network, also did push this session a lot. So I would just like to thank each and everyone. In fact, each and everyone working for the animals. And those are my last words, unless Swati and Dr. Priya have something to add, and then we can end the session. Mahesh, I'm so sorry, we are actually running out of time, so we can't take any questions or any comments. So over to you, Swati and Dr.
Thank you, Pratik. So, yeah, I really, really want to thank Dr. Kader and Dr. Akhtar for their time and consideration for this room. And of course, thank you so much, the entire Planet Earthling team, you, Pratik, Dr. Priya, and Global Jain Network. And of course, thank you everyone for your presence in this room. And yeah, over to you.
Yeah, I'll just say I really appreciate everyone in the room. I appreciate being able to learn about animal testing. I really appreciate everyone's time, particularly Dr. Kadir and Dr. Akhtar. It's been so educational, and I appreciate the fact that for you guys, it really is that it is about animals. I mean, yes, of course, it's about good science, but I appreciate that we have an organization that exists where the leaders see non-human persons as persons. So thank you.
Thank you so much. Thank you everyone for joining the call. And yep, we're up to do this again next year.
Wow,
Thank you so much. Take care.